Now, let me introduce you to Dr. Qun-Ying Yue. She is a medical doctor and senior pharmacovigilance expert at the Uppsala Monitoring Center. Prior to joining the Uppsala Monitoring Center, Dr. Ying worked as a senior expert at the Swedish Medical Products Agency, where, among other responsibilities, she served as a member of the Pharmacovigilance Risk Assessment Committee for many years. She's a specialist in clinical pharmacology and holds a PhD, and she is an associate professorship at Karolinska Institute. Dr. Ying will present her conference entitled Pharmacogenomics in Pharmacovigilance. We invite you to actively participate during this presentation. If you have any questions, please ask them in the chat on the left. We'll be responding to as many as we can during the allotted time. The rest of the questions will be answered and published in our Q&A section within Patient Safety Day on our webpage, isoponline.org. Without further preamble, please give a warm welcome to Dr. Ying.
Thank you. It's my pleasure to talk at the World Patient Safety Day today. The title of my presentation is Pharmacogenomics in Pharmacovigilance, Genomic Associations with ADRs, Adverse Drug Reactions. Here is the presentation outline. I will introduce the definition of pharmacogenomics, pharmacovigilance. I will explain the questions of pharmacogenomics in pharmacovigilance through some examples. At the end of my talk, I will briefly introduce you the pharmacogenomic special interest group within ISoP. Now, what is pharmacogenomics? The term pharmacogenomics sometimes is used interchangeably with another term, pharmacogenetics, which is the study of inter-individual variations in DNA sequence related to drug disposition in pharmacokinetics or drug action in pharmacodynamics that can influence clinical response. Pharmacogenomics is a broader concept, which is the application of genomic technologies to elucidate disease susceptibility, drug discovery, pharmacological function, drug disposition, and therapeutic response. What is pharmacovigilance then? Pharmacovigilance is a pharmacological science related to the collection, detection, assessment, monitoring, and prevention of adverse effects with pharmaceutical products. So the focus is on adverse drug reactions, which is any response to a drug which is noxious and unintended. It was previously referred to reactions that occurs at doses normally used. However, more recently, it also includes reactions that may arise from the use outside the terms of marketing authorization, which includes off-label use, overdose, misuse, and medication errors. So now, the variability in drug responses. When the same dose of a drug is given to different individuals, the effect will be different due to a number of factors. For example, physiological factors such as age, gender, pregnancy status, or pathological factors like diseases, renal diseases, liver diseases, or heart disease, etc. The environmental factors like smoking status, consumption of alcohol, or diet, or drugs, drug-drug interactions. Last but not least, genetic factors that will not affect the inter-individual variability, but also influence the interethnic variations. So what are the questions for pharmacogenomics in pharmacovigilance? Well, it's about biomarker. It's about use of genomic biomarkers to help with the patient selection, find the right patient, to help with the drug choice, to find the right drug, and to help with the drug dosing, the right doses. Sometimes it helps with the treatment recommendation, as how to monitor a patient. There are mainly two kinds of genomic biomarkers. One is to define the risk status for adverse drug reactions, and another is to define the rate of drug metabolism. There are also other biomarkers for disease diagnosis or disease prognosis, for example, in oncological implications or HIV infections. But today we are going to focus on the two genomic biomarkers in the context of pharmacovigilance. For the biomarkers to define risk status for ADRs, there are specific genomic biomarkers, genetic allele, for example, the HLA B5701 allele. In the case of a specific drug, abacavir, with hypersensitivity reactions. So this type of biomarkers usually is about immunological related adverse drug reactions and HLA alleles with a specific drug. Regarding the biomarkers to define rate of drug metabolism, this is not as specific as for the risk status for adverse drug reaction biomarkers. But these enzymes like CYP2D6, 2C9, 2C19, they are polymorphic, and there are variations in the genes that are coding these drug metabolizing enzymes. And all the dose-dependent adverse drug reactions will be increased in patients carrying these variations. We will come back to use CYP2D6 as a special example to illustrate this type of biomarkers. A few words on the genetic testing of the individual to define the risk status for ADRs. Here we need a specific drug, a specific gene allele, a specific ADR, adverse drug reaction, and we want to know in which population as well. For abacavir hypersensitivity associated with B5701 allele, the recommendation applied to all populations. With the same gene allele 5701, if you take flucloxacillin, the adverse drug reaction concerned is different. It's a liver injury in this case. For the drug of carbamazepine, if you are a Chinese or Thai individual, the risk for Stevens-Johnson severe skin reactions will be increased if you carry the HLA B1502 allele. However, if you are European descent and Japanese individual and take carbamazepine and have the gene allele HLA A3101, you will have increased risk for all kinds of skin reactions, not only the severe Stevens-Johnson syndrome. So that needs specificity for ethnic origin as well. And for the same adverse reaction for phenytoin, you have the same gene allele as for carbamazepine. But for allopurinol, it's another gene allele. So there are specificities for the gene allele, for the drug, and for the adverse drug reaction. Now, regarding the genetic polymorphisms in drug metabolism, there are additional aspects we want to understand. We want to know the substrates, the material or substance on which the enzyme acts on. We want to know the test methods, if it's phenotyping or genotyping. And we want to know if there are inhibitors or inducers. And we want to know the clinical implications. So for the 2D6 polymorphism, it was also called dibrisoquine dehydrogenase when it was discovered in 1970s. This antihypertensive drug, dibrisoquine, was metabolized to 4-hydroxydibrisoquine, and there are large inter-individual variability in this metabolism, which was discovered. The enzyme is called CYP2D6. To test this 2D6 with a phenotyping method, you need drug intake, need urine collection, for example, and analysis of the urine, and then you calculate a metabolic ratio with the parent drug dibrisoquine divided by the metabolite for hydroxydibrisoquine. This plot shows the metabolic ratio distribution. The higher the metabolic ratio is, the lower the enzyme activity will be. There is an anti-mode at 12.6 of the metabolic ratio. To the right, with the black bar, they are the poor metabolizers. They have a very high metabolic ratio, but very low enzyme activity. And for individuals with a metabolic ratio between 1 and 10, they are the intermediate metabolizers. Most of the subjects have a metabolic ratio between 0.1 and 1, and they are the extensive metabolizers. There are few individuals have a metabolic ratio below 0.1, and these are the ultra-rapid metabolizers. You may do a panel study in a group of poor metabolizers and compare with a group of extensive metabolizers. In this case, after taking haloperidol, you look at the plasma concentration, you find a significant difference between the two groups, indicating the involvement of CYP2D6 enzyme. There are good studies, well designed, to illustrate clinical implications. This study was done in the UK with a drug called Praxilin. It's an anti-angina product, which is not on the market anymore because of the neuropathic adverse drug reactions. This study was done in 3 groups of subjects. One is patients on the drug with neuropathic adverse drug reactions. The second group is patient on the drug without adverse drug reactions. And the third group is the general population without on the drug. What you see is the median dibrisoquine metabolic ratio was very high in patients with adverse drug reactions, 14.4. And in patients without adverse drug reactions, the metabolic ratio was at the same level as in general population. And what is very interesting is that if you look at the dose of Praxilin, gram per week, it's even lower compared with patient group without adverse drug reactions. And this is because the poor metabolizers, poor metabolism in patients group compared with the patient group with adverse drug reactions compared with the patients without adverse drug reactions. And 50% of the patient group with adverse drug reactions are poor metabolizers with metabolic ratio above 12.6. So this illustrates the importance for individualized dosing to find the right dose in right patient. Here shows the substrate of 2D6. There are beta blockers, antidepressants, antiarrhythmics, neuroleptics, and also drugs like analgesic products, and even anti-cancer drugs like tamoxifen, and also a relatively new drug, alleles, which is used for rare disease, Gaucher disease. We also want to know the inhibitors. There are potent inhibitors of the enzyme 2D6. After a single dose, 200 milligrams of quinidine, this could transform the rapid metabolizers or extensive metabolizers into poor metabolizers. Here shows some of the strong or potent inhibitors of 2D6, quinidine, fluoxetine, paroxetine, cinacalcet, bupropion. All of these should be avoided if you take tamoxifen for the treatment of a patient with breast cancer, because tamoxifen is activated by 2D6 to form the active metabolite. If the enzyme is inhibited, we will not have sufficient level of the active metabolite. The consequence will be the failure for tamoxifen treatment for this very serious indication, breast cancer. Another method for testing the polymorphism is genotype. We look at the DNA, for example, from a blood. And here we have to know the validity and utility of the analysis, and also the clinical validity and utility, meaning that the analysis should show the right results and to identify the right individuals, not misclassify them, and it should be feasible to do the testing, not for a drug which is used in acute situation. This picture shows the 4 genotypes of 2D6 polymorphism. To the left, you see the ultra-rapid metabolizers who carry more than 2 normal genes. And next to it are the extensive metabolizers who carry 2 alleles of normal genes, or the wild type genes, one from the mother, one from the father. And then for individuals with a metabolic ratio between 1 and 10, they are the intermediate metabolizers with a 1 active gene, 1 inactive gene. And in the poor metabolizers, they are homozygote for the mutations with a variant gene. This shows beautifully the gene dose effect. In 5 groups of patients with poor metabolizers or extensive metabolizers, intermediate metabolizers, and ultra-rapid metabolizers, you'll find the poor metabolizers with very high concentration of Nortriptyline, the parent drug, and very low concentration of 10-hydroxynortriptyline. And the opposite is true for the ultra-rapid metabolizers. And other genotypes are located in between. So far about inter-individual variability. Now, let's look at the interethnic differences in 2D6. Here shows 2 populations, the Chinese and the Swedish Caucasians. You see 2 differences. The first one is there is lower frequency of poor metabolizers in Chinese compared with the Swedish. And another difference is the right shift in the peak in the Chinese compared with Swedish. Why is that? You see the variant alleles, CYP2D6*4, that has no enzyme activity, and allele frequency is 12 to 21 in Caucasians. That gives rise about 7 to 8% of the poor metabolizers. And allele frequency in Chinese is only 1%. So this explains the low poor metabolizer frequency in Chinese. Then you look at the CYP2D6*10, which is associated with reduced enzyme activity. And allele frequency in Asia, in Chinese population, is 51%. And in Caucasians, is only 1 to 2%. And this explains the right shift of the metabolic ratio in Chinese. And in Africa, you see the 2D6*2XN that represent ultra-rapid metabolism, and you'll find higher allele frequency in Black Africans. So what is the clinical implication for interethnic differences? Suppose we have two populations, A and B. Although the inter-individual variability within A and within B are much bigger compared with the average differences between population A and population B, still, there can be important clinical implications. Suppose the rate of clearance of a drug below which there will be serious adverse drug reaction, and the frequency for this reaction to occur in population A, for example, 1%, and in population B, it will be 8 times higher. So this will be obvious to look at the frequency of a dose-dependent or concentration-dependent adverse drug reaction. This is a publication from 1990s in the US. Following 1 milligram dose of haloperidol, you see there's a large variability in haloperidol concentration in the blood. And you see the white bar representing Asian patients tend to have a much higher concentration compared with the black bars, Caucasian population. So psychiatrist tends to give lower dose to Asian patients in that hospital. So what do we find in the labeling regarding genomic testing information? There are 3 tiers of information. One is mandatory for the testing. One is recommendation, could be strong recommendation, could be weaker recommendation. And for information, when the data for recommendation is insufficient to make. So here is an example of product information regarding genetic biomarker testing. There are 2 examples where genomic testing is mandatory. The first example is abacavir, which is indicated in patients with HIV infection. And before treatment, all patients need to have screening for the HLA-B 5701 allele. And the drug can only be used in patients without carrying this allele. So this helps to choose right patient, right drug. In the case of Sadarga, which is eliglustat, precocious disease, this is indicated for long-term treatment who are 2D6 poor metabolizers or intermediate metabolizers or extensive metabolizers. And this biomarker helps the right dosing. You give 84 milligram twice daily in immediate metabolizers and extensive metabolizers, and you gave only once daily in poor metabolizers. And the drug should not be used in ultra-rapid metabolizers or in subjects who are indeterminate metabolizers, because the right dose have not been determined. Here are some examples for recommendation using the genetic biomarkers. For carbamazepine, the recommendation is quite strong. It says HLA-B1502 allele in Han Chinese or Thai population, genotype should be done whenever possible. And for other situations like allopurinol and skin reactions and simvastatin for the SLCO1B1 allele, the protein transporter, the recommendation is somewhat weaker. And this is based on the available evidence supporting the genotyping recommendations. There are also occasions that the information is, the labeling is for information, where it's not possible to give specific detailed recommendation for patient selection or for dose recommendation. In the case of tamoxifen, although it's not possible to dose patients according to genotype, it does contain information, very clear information, saying that potent inhibitors of 2D6 should, whenever possible, be avoided during tamoxifen treatment. As mentioned, the evidence for the mandatory genotyping or testing, it's usually derived from well-designed prospective clinical trials with high positive predict value and high negative predict value. And this is also illustrated for the case of carbamazepine. And the evidence looks different, and the recommendation in the labeling also look different. So now I would like to mention the ISoP Pharmacogenomics, this special interest group, which is just established in May this year. We know there are large variability in drug response related to genomic variations. And we know there are subset of patients with a different benefit risk profile. And we know that at the drug approval, there are limited information for genomic subpopulations because the studies usually limited in size, in number, and the patients with rare variations will not be discovered during the clinical trials. So the pharmacogenomic special interest group, SIG, has the aim to provide opportunity for ISoP members interested in pharmacogenetics, genomics, to share information and to support pharmacovigilance relevant to the medicinal products with pharmacogenomic associations. For those who are interested, could go to the ISoP website and to have more information. This is my last slide, a few words about future. Regarding clinical recommendations, as we mentioned already, it's not always possible to have clear clinical recommendations when you need it, because the studies or results available derived from healthy volunteers, limited numbers, single dose, not possible to guide clinical use with specific dosing according to testing. But this could be changed. With the example alleglustart, you could, through the biomarker use, to dosing the patient very specifically in patients with the different genotypes or phenotypes. So in the future, with pharmacogenetic-based patient-tailored drug therapy, more prevention-directed will be feasible. Thank you very much.
Dr. Jin, thank you so much for your very interesting presentation about pharmacogenomics and pharmacovigilance. We have some questions from our attendees. So our first question is, now we understand that there are many examples where the adverse drug reactions are due to genetic variants in the patients. How do we discover an association between a genetic variant and an adverse drug reaction?
Thank you for the question. Well, many serious adverse drug reactions, in particular the idiosyncratic immunological reactions, as we already mentioned, may not be discovered in the clinical trials. It may only be discovered late in drug development or in post-marketing phase. It's therefore very important to be alert with pharmacovigilance activities. As a patient, healthcare professionals to report the adverse drug reactions and to collect this information, stored information into databases and detect signals, and also do research. One possibility to discover association between the genetic variant and an adverse drug reaction is to collect genetic materials, such as DNA, from blood of a patient who have suffered from serious adverse drug reactions, and then to have them in a biobank, for example, and then use this genomic technology, for example, a GWAS study, a genome-wide association studies, to find any differences, if there are, in genes between these patients and the controls. And in this way, to compare patients who do not have the reactions, and you identify a particular allele that stands out. In Sweden, there is a project called SweetGene, which is a collaboration between academy and regulatory authority. And DNA samples from patients with different adverse drug reactions with some common drugs and serious reactions, they can be collected and analyzed. And this is a good way of performing researches, because serious adverse drug reactions are usually rare, and you will need international collaboration. And I think the ISoP specific group may facilitate this. Thank you for asking.
Great. Our second question is, how do we know whether the genomic testing requested in the label has any impact on the risk of the adverse reactions?
Oh, good question. As we mentioned that when the genomic testing is mandatory, there must already be good evidence supporting the association between the biomarker and the ADR. But in addition, the impact of required genomic testing should be monitored, and it can be monitored. There has been for example, in the occurrence of a serious skin reaction associated with the Carbamazepine, which has been studied by a group of researchers in Chinese population. They compared before and after the patients being tested. It was found that the pre-screening of this allele, HLA-B1502, and avoiding Carbamazepine in the patients who carry this gene, and the risk has been diminished almost, and there were no cases in the exposed patients who do not carry this allele. And there are expected to be a number of cases based on historical statistical figures. So this shows that it is effective with testing, screening patients before starting treatment. There were also other examples that show benefit of the genotyping test. For example, in the case of abacavir, the sales of this drug decreased a lot due to the hypersensitivity reaction. But mandatory testing, putting this in the label, and the risk has also diminished, minimized, and the drug has been saved by genotyping test. Thank you.
Thank you so much for your answers, Dr. Ying. We have a lot of questions, but due to the schedule, the rest of the question will be addressed at the web page. Thank you so much again for your great presentation.